Hi, I'm Gbola Amusa. It's my pleasure to introduce our next guests from Solid Biosciences, Joel Schneider, who's Chief Operating Officer, and Louise Perry, SVP of Technical Operations. The format for this session is a roughly 40-minute presentation and fireside chat. If you have any questions during the session, feel free to type them into the question box at the bottom, and I'll do my best to ask on your behalf. Joel, Louise, thank you for joining virtually today, and please start your presentation.
Great. Good morning, Gbola, and thank you for hosting us. And I'm really pleased to be here today with our Head of Technical Operations and our SVP, Louise Perry, to discuss our manufacturing platform. And the conversation and presentation that I'll provide is really through the lens of our lead program called SGT-001 and the iterative, careful process development that we've done over the last 6.5 years to get us to the point that we're at today. I think we've learned a lot of lessons, and we're really excited to share with you our view of AAV manufacturing through the lens of Solid's perspective. I guess I can skip over my forward-looking statements. As I just highlighted, today's conversation and the slides that I've put together with Louise are really presented through the lens of SGT-001. And as a reminder, SGT-001 is the vector that we're currently testing in the IGNITE-DMD clinical trial. And by vector, I'm referring to our novel microdystrophin transgene for application in Duchenne muscular dystrophy. It refers to the promoter that we're using to drive expression of our microdystrophin transgene called CK8. And we're using the AAV9 classical derived capsid, which has shown high tropism for skeletal and cardiac tissue. As a reminder, our microdystrophin is unique because of certain domains that we've chosen to include. And we've now shown clinical differentiation of our microdystrophin through the lens of the incorporation of this nitric oxide synthase binding domain, which adds additional functionality to the microdystrophin transgene. And as a reminder, in March, we shared for the first time clinical data from the ongoing IGNITE-DMD clinical trial, which highlighted that at both doses administered, 3 patients at the low dose and 3 patients at the high dose, and with over a year of follow-up in all of those patients, signs of clinical benefit. Now, as I've mentioned, we've learned a lot about AAV manufacturing through the lens of this program, through SGT-001. When we started our iterative process development over 6 years ago at this point, we chose to select a fairly novel and differentiated manufacturing platform that we believed could successfully be scaled to the needs of the Duchenne muscular dystrophy community, could be tech transferred, and we'll talk more about that in the Q&A, to multiple CDMOs for production, and offered versatility in a platform that allowed us to both continue scale up and scale out, meaning not just the volume of bioreactor production, but also the ability to chain multiple bioreactors together for continuity and processivity of our AAV production. Equally as important, we chose to scale a manufacturing platform that uses proven and validated standard bioreactor systems, which really is important, as I mentioned before, for thinking about tech transfer and successful implementation at multiple sites. Now, we've taken this process from a very small-scale adherent platform, actually even before cell stacks, although I know this slide really highlights it initiated cell stacks, and carefully performed the process development internally at Solid, leveraging our core capabilities in process analytical development, as well as our manufacturing science and technology teams, to scale this process to the volume that we're currently operating at for the IGNITE-DMD clinical trial. And that's the 250-liter scale. And as we get into the fireside chat and the Q&A, Louise will highlight for you some of the strengths and some of the observations that we've made as we've scaled this platform around the equivalence that we've largely seen as we've scaled up the process. Now, we highlighted in March, as we shared data in IGNITE-DMD, that we're continuing to do process development and scale up of this platform to really leverage the needs of a phase 3 study and ultimately potentially a commercial gene therapy product. And the data that we've generated so far on our scale up suggests that this platform does have the capability to scale to the examples highlighted on this slide to 500 liters and beyond. But equally, as I mentioned, we believe there's a lot of strength in the versatility of not just thinking about scale up, but also scale out and the chaining of multiple bioreactory systems as well. We've been working on our manufacturing platform for over 6 years now, and we initiated that process development, as highlighted on this slide, in 2016, give or take. We initiated that process development in close collaboration with the CDMO, although early on initiated our own process development build-outs so that we could really control what we viewed as critical parts of the process development, the scale-up efforts, as well as the critical aspects of analytical development related to release and characterization specifications. And I'll touch on that in a moment. We produced our first GMP material in 2017 using what we now consider a first-generation AAV manufacturing platform, and that was at the scale of 250 liters. And so importantly, every patient that has been dosed in the Ignite DMD clinical trial has been dosed at the scale of 250 liters. In 2018, as we were producing at the GMP scale for Ignite DMD, we began to establish and initiate work on a second-generation AAV manufacturing platform, which we've now begun to utilize in the clinic in the Ignite DMD trial. And when I refer to a second-generation AAV process, I'm not just referring to the AAV production, and I'll talk about that in just a moment. I'm also referring to the development of novel analytics, tightening of specifications, all of which I believe are some of the most interesting areas of core technological development in AAV manufacturing. So to summarize that, in association with the development of key new attributes of our second generation AAV manufacturing platform, we also were in parallel refining all of our analytical capabilities, refining our specifications for release and characterization criteria, and that includes the introduction of an in vitro expression assay. There's a lot of talk today around potency and infectivity. We spent a lot of time developing an in vitro potency platform, an expression platform that allows us to use relevant cell lines to detect and assess the relative potency through microdystrophin expression in cell lines. And what that offers us is a highly quantitative, high throughput method for releasing materials. So we developed these second generation processes. We improved our manufacturing methodologies. We produced AAV GMP materials using these second generation processes, which were key for the demonstrations of comparability back to the first generation process. We submitted an IND, and we're now in the clinic with the second generation method. And I think there's an adage that's been around for a long time, and it came along well before AAV manufacturing, this notion of the process of AAV manufacture really defining the product. And what you can see on this slide highlighted in a representative fashion are all the steps that go into AAV manufacturing. That includes the earliest stages of cell thaw for the thaw and the cell culture associated with the cell line of choice that produces your AAV, the actual AAV production in your bioreactors, the purification downstream of those materials once they're produced at the upstream step, and then finally your fill and finish. And what you can see going from left to right on this slide is that every step of this process is highly controlled. And there are in-process monitoring steps, in-process analytics that allow us to understand how the bioreactors, how the cell culture is operating, things around viability, productivity, pH levels in our systems. All of this is carefully controlled and monitored, and all of the specifications that we use for in-process monitoring have been carefully developed by our internal process development and analytical teams in close collaboration with manufacturing science and technology. We also have a battery of in-process testing that we run at each step of our manufacturing process. That's highlighted as you shift over from in-process monitoring to in-process testing. And then finally, as I mentioned, we've carefully developed our analytical development criteria, our acceptance criteria for successful release of our materials. And I think what we've seen over the last 6 to 7 years of AAV process development is that acceptance criteria and the analytical tools are really matching the evolution of a very nascent field of manufacturing. So just as we're really scaling and learning more about gene therapy manufacturing, it seems like on a daily basis, the utility of the analytical tools that we have at our disposal to really understand our AAV product the requests are being elevated in the eyes of the FDA, and it's our responsibility as sponsors to really match the technological capabilities with the demands of the regulatory agencies. So really, in summary, as we've scaled our process and as we've begun to produce materials using our second generation process, and before that, our first generation process, we've carefully iterated and improved on all of our capabilities, internal to solid, that allow us to really understand our SGT-001 and subsequent pipeline assets as well. One key criteria of our second generation manufacturing process was the implementation of a scalable purification step that allows us to remove the majority of empty capsids from our drug product. And that's highlighted here in representative images, a comparison between our first generation process, which really produced out of the bioreactor without any specific purification, a mix of 50% full and 50% empty capsids. We've chosen to implement this purification step that eliminates the majority of capsids and gets us to over 90% full on average in order to reduce the total viral load that any patient gets when they receive SGT-001. And the impact that this purification step has on the total capsid load, if you will, is represented on the right-hand side of this slide. And specifically what it means is that for any given target vector dose, we've effectively reduced the total viral load in about half. It's about a 47% mean reduction in the total viral load that any patient gets utilizing our second generation process. I just want to take a step back and highlight, as I mentioned on our timeline chronography slide, this has been a process that our internal PD team has been working on for a few years before we chose to implement it at the GMP scale. And it was the careful analytical tools that we developed that allowed us to produce this material at the GMP scale and then demonstrate comparability through statistical analysis using all of our analytical attributes to demonstrate comparability between the second generation process and the first generation process, meaning that in the eyes of our release and specification criteria, the materials were really only different in the way that we want them to be, specifically in the reduction in the total capsid load, as well as a few other steps that we chose to implement as well. And so in summary, and to allow us to give some time to have a really nice Q&A, we have built at Solid over the last 6.5 years a scalable manufacturing platform for the production of AAV vectors. We can achieve high yields with a high percentage of full capsids due to the implementation of our second generation manufacturing process. And this process is very well suited for scale up in commonly used suspension bioreactor platforms. The novelty of our platform using the herpes simplex virus production method gives us total control over the supply chain. And I'm sure Louise will be happy to talk to you more about that in the Q&A section. We continue to refine the HSV platform. We're improving our purification strategies to continuously refine and optimize our product quality as we think about a phase 3 study and commercial timelines. We've also continued to optimize our AAV productivity of the HSV process in of itself. And certain steps that we've made in the manufacturing process, we believe, will over time allow us to reduce the time of GMP production to ultimate release. As I've highlighted fairly extensively, the AAV production side is really only half the story. We've built very strong core capabilities in analytical and process development, which we believe are a vital and strategic foundation for success in gene therapy manufacturing. And that expertise and know-how are very critical as Solid begins to think about assets beyond just SGT-001. And we've built really deep expertise in the development of characterization and release assays due to the strong core of scientists that we have in process development and analytical development, which makes up a majority of Solid's team. And then finally, we're continuing to refine our process, and there are additional improvements planned and underway for phase 3 production as well as beyond. And so we really expect to continue to streamline production as we move forward. So let me stop there, Gbola, and hand it back to you. But thank you for the time to present.
Absolutely. Yeah, thanks, Joel. That last slide was a great summary of your manufacturing processes. But I'm interested in how your approach differs in terms of CMC capabilities and strategy versus the rest of the cell and gene therapy space. So what are the advantages? And I know you mentioned that you have total control via HSV. So what does that mean, and what are the other advantages?
Great. So perhaps I'll just highlight the core science behind our AAV manufacturing platform and then hand it to Louise to talk to you about the manufacturing side of it and what we believe are some core differentiators. But fundamentally, as you highlighted, Gbola, the process that we're using is somewhat distinct from many other players in the space, although there are others using the HSV platform. The major differentiator is the starting material. We have 2 different herpes viruses, one containing our gene of interest, in this case, our unique nNOS containing microdystrophin, and the other containing the sequence necessary for producing the viral capsid, the AAV9 capsid. So rather than using plasmid-based production or plasmid-based transfection and in our suspension 293 cell line, we're using these 2 HSV viruses at a certain stoichiometry to co-infect our suspension production cell line to then produce our AAV. And all of that production, both the HSV production and the AV production, is something that Solid has control over. But let me hand it to Louise to talk to you a little more about those core differentiations of the technology. So, Louise?
Thank you, Joel. As Joel highlighted, our HSV process allows us to keep control over our starting materials. A lot of companies need to order their plasmids and get in line and wait, and this can affect their ability to produce AAV. For us, we start with a master viral bank, one for the GOI and one for RepCap, and can then from this starting material produce our HSV, which are the helper viruses, which will produce AAV. So we can maintain a known stock quantity of HSV and then as necessary move forward with our AAV process. We've been improving, as Joel highlighted, not only our AAV process to improve purity and quality, but also we've been working on our HSV process as well, so that we over time, and as Joel also highlighted, analytics and the evolution of our analytics have allowed us to improve the quality of our HSV as well as the quantity that we're able to produce. And we're actively working on continuing to improve that process to ensure that we have starting material that will then allow us to, over time, grow beyond 250 liters, continue to scale up to 500 liters and maybe even to 2,000. We've seen very good conservation of yield from 10 liter up to 250 liter. It gives us good confidence that we'll maintain that yield as we move up in volume. Joel also highlighted that we're not only looking at moving up in volume, we're looking at moving up in quantity of bioreactors, so scaling out. And even before we move to a 500 liter, we're looking at running two 250 liter bioreactors simultaneously, and then being able to purify larger quantities of material through to DP.
I think one of the interesting differentiators also of the HSC platform, and it was highlighted on one of our slides, is that upstream, without any sort of specific purification, we do seem to see a slightly higher level, a full to empty ratio. In other words, there seem to be more capsids that are full than perhaps with other methods. And I'll just highlight that I think we looked very early on at the landscape of manufacturing approaches, and given Solid's unique mission and our very focus on trying to move forward with the technologies we felt could be most meaningfully applied to Duchenne, we felt that some of these unique advantages, like the HSV starting material, would inherently allow us to have a platform with a significant amount of versatility.
Right. So back to the discussed theme of manufacturing defining the product. I think Solid, in theory, could become one of the best case studies of how a leading process can enhance the product's attributes. Obviously, more work and data is needed, but can you give an overview of your clinical mitigation strategy and how you've improved your manufacturing process?
Sure. I'll cover the first half and then hand it to Louise to talk to you a little about specifically the changes that we've made. But the primary changes in Ignite DMD were really synergistic in nature. On one hand, it was a revised clinical strategy and these critical manufacturing changes, which we view as equally as important. On the clinical mitigation side, we have chosen to dose a number of patients at a lower weight, as well as the prophylactic use of two complement inhibitors, eculizumab and a C1 esterase inhibitor. We've also, and that goes along with this notion of the total viral load, leading to a robust activation of the immune system, which many sponsors in the space have seen this level of complement activation or innate response. We've also increased very transiently the dose of steroids, the prednisone dose that we're providing, during the first month of dosing. And that's really the clinical strategy side to kind of mirror this notion of the total viral load being an important part of the aspect. But let me hand it to Louise to talk about the specific manufacturing changes and why we think that they're unique in some of their scalability aspects. But Louise?
We implemented purification technology to eliminate the vast majority of empty capsid in the downstream process. And the way that we did this, there are multiple opportunities for removal of empty capsid, but we found it really important not only to be removing empty capsid, but to make a scalable process. As I was highlighting earlier, we intend to grow in terms of the volume that we'll be producing per bioreactor, as well as the number of bioreactors that we intend to run. And then we'll be doing a single purification. So that purification technology needs to be scalable so that we can scale up very quickly and easily to keep up with the scale of the upstream technologies. So that's a very important piece of what we've done. We've also shown that over multiple batches in GMP production, we've been able to achieve approximately 90% full capsids consistently. So we see a very consistent and repeatable process with that downstream operation that's been added. We've also seen very tight control of our quality aspects. So to our points earlier, as our analytics have grown and developed over time, we've been able to even more tightly control and better understand how every change that we make as we continuously improve our product also changes, shifts, and improves our quality at the end of the production.
Got it. So I think the conventional wisdom is that making AAV is a bit more complicated than making other vectors, such as adenoviral and lentiviral vectors. What's your take on that issue, and what are the advantages of AAV generally, and maybe solids AAV9 specifically?
Let me hand that one to Louise. I'm excited for her to answer that, given her long history in the biologics manufacturing space. So, Louise, let me hand it to you.
Lots of folks have come into this with a long history of protein manufacturing and biologics manufacturing. And I think one of the big things that's happening is that we're gaining experience. So this is all new, or nascent, as Joel likes to say. And every time we run a production, we learn something, right? So we learn something every time we run a small scale in PD. We learn something every time we run a larger scale production process. And we take that learning and we apply it as we move forward. Eventually, we are going to have to lock down the process, right? We're going to, our continuous improvements will need to end. We'll need to lock a process. We'll need to be able to validate that process. But the more we know and the more we understand about how the controls that we implement and manage during production affect our final product, the better off we will be as we go into the validation state.
I think one of the interesting aspects as well, of AAV9 specifically from a scientific perspective, is that and this is not unique to Solid, AAV9 does seem to be a fairly good quote unquote producer. In other words, that when we compare its productivity to other capsids, it does seem to have a fairly good overall productivity. And I think that's an important part, taking this back to the process development side. Every vector is different. And by vector, I mean capsid, transgene, promoter. They all have different criteria. And while certainly the lessons and experiences apply from program to program, they all have different attributes around productivity, around yield. And AAV9 does seem to be a fairly good producer, which certainly works to our benefit.
And then back to the issue of full versus empty capsids in the manufacturing process. I think your slide showed that you've gone from 50% full to 90%. So can you talk about how that's accomplished and where you can actually go? Like what's the max that can be achieved with your processes? Or alternatively, are you refining your process more, and where do you hope to go? Where do you aspire to go?
It's a great question. It's always good to be aspirational. I think that fundamentally the reason that we highlighted the improvements in purification and specifically this overall average of 90% is because in our hands, and specifically as sponsors, we believe is an important part of our strategy for moving the program forward. And I think every sponsor has their own experience from candidate selection through IND enabling efforts, through clinical experience that drive whether or not there's a rationale for elimination of empty capsids or not. And many sponsors choose to eliminate, many don't. It's really driven by everyone's independent experience. I think for us, and Louise really highlighted well, the adaptation of a scalable purification step to eliminate these capsids, we think is certainly an important cornerstone of our manufacturing process. And I think over the years, there have been many different methods utilized for elimination of empty capsids. Some more challenging, although nothing is impossible, but some more challenging to implement at a larger scale than others. The technologies that we're using allow us to scale the purification as we go up in scale or as we go down, and we think that does add significant versatility. I can't tell you today whether or not we'll continue to try and eliminate more empty capsids. We think it's a pretty good spot to be in. But certainly, as we explore next generation capsids, which we presented some data on that in our recent corporate deck, we'll see how these capsids behave and whether or not the current purification strategies that we use for SGT-001 are amenable to application in some of these other programs as well. So today, we're very happy with, as Louise highlighted, that we're getting this consistent GMP scale enrichment or elimination of empty capsids. That for us is a very meaningful step. And all the work and comparability that went in to get there is an important cornerstone of our efforts. And we'll see where we go from here based on Solid's trajectory of SGT-001, as well as the next generation pipeline rollout.
Understood. So can you discuss the new clinical protocol that's being used to dose the next patient? So in addition to that, what are the ages of weights of the patients? How might that affect things as well?
Sure. Well, what we announced in March around the data disclosure that we shared highlighting benefit in these patients was that for the first couple of patients dosed with the second generation process and with the Reopen Ignite DMD study, that these patients would be below a weight range of 18 kilograms. And again, that was to just match the idea of reducing the total viral load. And I also highlighted that alongside the weight reduction, we are using this prophylaxis complement strategy, which we think is an important forward-looking way of treating high-dose, high-weight patients with systemic gene therapy. And we're going to continue to monitor and explore how that prophylaxis strategy is going and how it allows us to implement in future work. So for us, for the first couple of patients dosed, we're going to be restricting the weight, and then we'll work closely with our advisors, with our DSMB to think about next steps in the program. But a weight restriction, prophylaxis, using complement inhibitors, improved manufacturing strategy to reduce the total capsid load, and transient elevations in our immunosuppression strategy with prednisone.
Got it. So, all these updates, changes are meant to benefit the patient. That's the key thing here, which we can all agree upon. So what are you hoping to see in the new patients dosed in terms of microdystrophin expression? Yeah,
I think we're very happy with the expression and then the subsequent clinical benefit that we were able to share for the first time in March. We've shared for the first time functional data with some endpoints that have not yet been shared in gene therapy for Duchenne, specifically 6-minute walk data, as well as forced vital capacity, which is an incredibly important component to consider as you think about patients in an age range where motor function assessments on ambulation are simply not really going to be the right suitable endpoints. And so I think what we're looking for in our patients are similar signs of efficacy driven by similar levels of protein expression. In our comparability efforts, we demonstrated fairly in our in vitro expression systems, similar levels of protein expression. And I think equally, if not more important, signs of benefit, even early signs suggesting that, we're seeing what we saw in the first 6 patients in the form of a separation from the natural history of the disease, as well as potentially signs of improvement. But we've got more patients that we need to dose and more data that we need to assess. But we're certainly happy with what we saw. We expect the revised manufacturing process to produce a similar level of efficacy.
Got it. And then there's an inbound question on to what extent can you change your process, your product, et cetera, without having to start over? Is there any new understanding you've had about that question based on your interactions with the regulator in the last year or two?
Well, I'll give you a high-level version of that, and then Louise can certainly spend a few minutes talking you through what actually goes into the process of demonstrating comparability. But I think we've learned a massive amount about the analytical side of our production platform, as well as the process development side, and that's all core internal expertise that we've amassed. It's knowledge that you can only derive by experiencing AAV manufacturing. And I use the word nascent a lot, but it's very true. There's a lot of lessons that we've learned from the biologic space, and a lot of new ones that we've had to learn from the AAV side. And all of that went into our thoughts around the implementation of our second generation manufacturing method. So we've learned a lot, and I think that allows us to be versatile, not just in our manufacturing platform, but also versatile in our thoughts about how we continue to broaden our AAV capabilities. But let me hand it to Louise to talk a little bit more about what comparability really looks like, and what you would really have to do to change manufacturing platforms or continue to improve it in the eyes of in the FDA and really in the eyes of all of our stakeholders. So Louise, let me hand it to you.
It's an interesting job to show that when you make changes, you haven't changed the ultimate outcome of the final product. So we have many tests that we use as release criteria with specifications that have over time, we've been able to tighten. So over multiple batches of production, we look at each aspect, each attribute of the final product. And when we make changes along the way, we're looking not only at the final product, but also, as Joel highlighted earlier on in the slides, we have in-process technologies that we're also using. So analytics along the way, every step of the way, we're looking at, does this product look the same now in this new process as it did in a previous process? So it's a massive amount of documentation, of data collection, and of data analysis to ensure that all throughout the process, if there are changes, the changes we're looking for are improvements. So we need to understand with any particular parameter, what's good, what's moving in the correct direction, what's moving in a higher quality direction, and what's moving outside of the norm. And if we start to see things move outside of a normal range, then that indicates to us that we need to work more on that particular change to ensure that it's not going to change the outcome at the end of our whole production process. So really, it's from the manufacturing sciences and technology side, we're collecting data throughout the production process in GMP. And we're not only comparing that data to our previous GMP production, we're also comparing that data to small scale production to ensure that whatever change we made in small scale has conserved itself through scale up to our large scale processes. And analytics are really the way that we do that. We have to have good, tight, high quality analytics where we can show that the tests that we're running at small scale are comparable to the tests that we're running at large scale. And therefore we're able to, through analysis of that data, show comparability from one process to an improved process. And that all needs to be documented, written down, and supplied to the FDA.
Thanks for that. So it's a rapidly evolving field, AAV manufacturing. So what do you think the biggest advancements have been in recent years?
I'll give you like a high science version, and Louise, I'd love to hear your opinion as well. I think I said this before, and I mean it. I mean it very honestly. And if Brian Collins, our head of analytical development, is listening, it's going to inflate his ego a little bit more. But I do believe that what we're learning around analytics, the tools that we can use to really understand AAV and the capsid biology are just rapidly advancing, and it's matching or exceeding the technologies that we're developing on the manufacturing side of itself, so the bioreactor systems as well. But I think that there's, for one of the first, well, not one of the first times, but certainly at this point, we're at a juncture where the core technologies that we have at our disposal are matching the pace of the rapid clinical translation of these AAV gene therapies, and we can utilize that to really understand the types of changes and modifications we can make. And to really understand our drug product as we go through iterations of process development. So for me, I've been blown away by how fast the analytical side of this field is rapidly developing, and how the responsibilities on sponsors are continuously increasing to match that level of understanding. So for me, it's really around the analytics. But Louise?
I think it's experience. Everybody came into this field with no experience maybe 5 or 6 years ago, and we have been learning so much so quickly. That learning curve is still extremely steep. as we bring our backgrounds in biologics into this new field of AAV viral vector manufacturing, we're learning with every single production process we run, with every single small scale production that we run. And I think the way that we're going to continue moving forward is to continue to be open to learning and really understanding that the unexpected is going to crop up. And every time we get something that's unexpected, that's a massive opportunity for us to learn and be able to then utilize that learning for the next processes that we run as we're moving forward. And whether that's the next scale up that we do, or if it's the next generation process that's coming along, being able to really document, understand where those learnings are coming from, and then move forward, implement those learnings in a strategic way is one of the things that's been most important.
Got it. And what are the biggest CMC challenges currently from your perspectives?
Louise, please.
From my perspective, it's producing enough material to meet demand. And with Duchenne in particular, this is it's a large challenge. So we have to understand what affects our yields, understand how we can scale up as well as out in order to be able to meet demand. And going to a suspension process for us has been really key to being able to achieve that. It allows us to scale much more easily than if we were using an adherent process where our only opportunity would really be to scale out. In this case, we're able to scale both out and up. And, in talking to some other folks who are also, working in this field, supply is really one of the most challenging aspects of CMC.
I think the only other thing to add to that and to complement Louise's answer is that the rapid rate of translation in gene therapy from preclinical development to clinical proof of concept is incredible. I think back to when we started our gene therapy efforts, 6.5 years ago, and there were a few studies that were systemic, high dose studies exploring what this could look like, and it's incredibly encouraging and really rewarding to see this field rapidly accelerating. And so matching the excitement and the passion around clinical translation and proof of concept with the rigorous demands of a quality program and a quality manufacturing strategy is something that needs to be understood and is an important challenge, but it's also one that highlights just how successful the field's been in translation.
Understood. And then the final question, I'm always interested in the future, where the field's going. So what important innovations in manufacturing might actually enable your platform to meet or exceed its goals?
Louise, would you like to?
I think, again, the ability to scale up. And I think the other piece that really comes in is flexibility, right? So the more flexibility you have in manufacturing, the better off you are, because whatever you're presented with as a challenge, you can work your way around that challenge if you have some flexibility in maybe the raw materials that you use or the way that you operate. So understanding your platform as fully as possible really allows you that flexibility. So I think that will help to define where the manufacturing processes can go in the future.
Joel, anything to add?
No, that was a great answer.
Great. Thank you, Louise. Thank you, Joel.
Thank you, Gbola.
We now conclude this session.